Hello students, good afternoon. Today we are going to discuss about the gene mapping. Gene mapping. The gene mapping means what? It is the diagrammatic and the graphical representation. Diagrammatic and the graphical representation of the relative distance between linked genes. Means the gene mapping means the diagrammatic and graphical representation of the relative distance between linked genes. We already discussed about the linked genes. The linked genes means on the chromosome, when the genes they are closely placed on the chromosome, so they are known as the linked genes. Okay. So the gene mapping means it is a diagrammatic and graphical representation of the relative distance between the genes, okay, or between the linked genes. It is known as the gene mapping. Okay. So here we are going to discuss the example 2-point test cross. Okay. So test cross means we know that when the F1 progeny, when it is back crossed with the recessive parent, then it is known as the test cross. Okay. So F1 progeny which is heterozygous, when it is back crossed with the recessive parent, it is known as the test cross. Okay. So the test cross when the two characters or two genes or two linked genes, they are considered or studied at one time to calculate or to map the gene distance, okay, or to calculate the distance between two genes. Distance between two genes is called as 2-point test cross. It is known as 2-point test cross. The 2-point test cross means when F1 progeny back crossed with the recessive one, that is known as the test cross, and when two genes or two linked genes they consider or study at the same time or at one time to calculate the distance between these two genes, it is known as the 2-point test cross. Okay. So by mapping or by gene mapping we can calculate we can calculate the distance between two linked genes. Okay, we can calculate the distance between two linked genes. Okay, so here we are going to discuss one example. Okay, one example of 2-point test cross. So in corn plant there are the two parents. One is the purple and full. So the color of the endosperm, that is the purple, it is denoted by capital P, and the full it is denoted by the F, capital F. Okay. The second, that it is the dominant parent, pure line dominant. It is pure line dominant parent when it is crossed with the recessive, that is the white endosperm and the shrunken endosperm. Okay, so it is the recessive, pure line recessive. So white is denoted by the small p and shrunken by the small f. Okay, so when there is a cross between them, so the purple and the full, when it is crossed with the white and shrunken. Okay, so capital P, capital P, capital F, capital F, when it is crossed with small p, small p, small f, small f, it produces capital P, capital F, small p, small f. When they are crossed, it produces capital P, small p, capital F, small f. So it is purple and full. So it is the F1. Okay. So in 2-point test cross, now we have to calculate the distance between this purple and this full. So it is the gene mapping. So to calculate the distance between the purple and full, or the distance between the white and shrunken. Okay. So it is the 2-point test cross because the two characters are studied. This purple color and full. Okay. So that is the full shape. It may be full or it may be the shrunken. So it is the 2-point. Two characters are studied at the same time. That means the two genes, they are taken into consideration for the study, and now we have to calculate the distance between these two genes. Therefore it is the 2-point test cross. Now this F1, that is the F1 is purple full, is cross, it is test cross, therefore it is cross with the completely recessive parent, so white and shrunken. Okay, so this purple full, it is the F1. So capital P, small p, capital F, small f, and here the small p, small p, small f, small f. Okay, so it produces the 4 different types of gametes. So first, that is P, capital P, capital P, capital P, small p, then here capital F, small f, capital F, small f. We produce only one type, P and F. Okay, so they produce the progenies. Okay, and these progenies are the 4 types of progenies. Okay, so 1, 2, 3 and 4. So it is the purple and full, white and full. So it produces P, capital P, capital F, capital P, small f, small p, capital F, small p, small f. Okay. So first of all it produces capital P, small p, capital F, small f. That means it is purple and full. Okay. So this is, it is small p, it is capital P, small p, small f, small f. So it is purple and shrunken. Okay. Then here small p, capital F, small f. That means it is white and full. Okay. And small p, small f, small f. It is white and shrunken. So 4 types of parents are produced. That is the purple full. Okay. Then purple shrunken. Then white and full, and white and shrunken. So it is observed that this test cross, it gives the purple purple shrunken, it produces 60 progenies. Okay. White shrunken, it produces 1575 progenies. Purple full, it produces 1667 progenies, and white full, it produces 58 progenies. Okay. Now we have to calculate the distance between this P and F. Okay. So there are the parental combinations are more. Okay. Parental combinations are more. That is the purple full and white shrunken. And these two are the recombinant. Recombinant. And this recombinant, they are very less. Only 60 and 58. Okay. Now we will calculate the distance between the white and shrunken gene, that is the P and F, or purple and full gene. Distance between purple and full. Okay. So how can write the answer? So the total number of progenies. Okay. The total number of progenies. Total number of progeny. Okay. So 1575. Okay. So these are the white shrunken plus 58. These 58 are the white and full. Then 60 purple and shrunken. And the purple and full 1667. So this total, the total of all these four, it is 3360. Okay. And how many the recombinants? The number of recombinant. Number of recombinants. How many recombinants? The 58 plus 60 means it is only 118. Okay. Now we have to calculate the crossing over frequency. So the crossing over frequency is equal to number of recombinant. That means non-parental types divided by the total number of progeny. Total number of progeny obtained in test cross. Obtained in test cross. Okay. Into 100. Into 100. Now see here. The recombinants are 118. And the total number of progeny 3360 into 100. So it gives the answer 3.5%. So the percentage of crossing over frequency is only 3.5. Okay. So the distance between P and F, or the chances of the percentage of the crossing over between the purple and full is 3.5%. Okay. So according to Morgan, according to T.H. Morgan, according to T.H. Morgan, the 1% crossing over is equal to 1 unit. is equal to 1 unit of distance. 1 unit of distance between the linked genes. Okay, and that distance is calculated by the 1 Morgan unit or 1 centimeter. Okay, so here in this test cross, what is the percentage of crossing over? That is 3.5%. That means the distance is 3.5 Morgan unit, okay, or 3.5 centimorgan. This cm, it is not the centimeter, it is the centimorgan. Okay, so the distance between this purple and full, the purple and the full, or we can draw like this, it is the chromosomes here, the purple is present and it is the full is present. So the distance between this purple and full is 3.5 Morgan unit or 3.5 centimorgan. Okay, so it is the distance between this P and M. So they are closely placed and therefore there are less recombinants and more parental combinants. So there are the chances of only 3.5% of crossing over between the purple and full. So it is a 2-point test cross. Okay. So 2-point test cross. Gene mapping means what? Today we have discussed about the gene mapping. The gene mapping means it is the diagrammatic and graphical representation of the relative distance between the two linked genes. We have discussed the 2-point test cross, and the 2-point test cross is also known as the dihybrid test cross. It involves the test cross between two linked genes at a time. Okay. So in this example, we have discussed the white, it is the corn plant, corn means maize plant, which have the purple endosperm and the white endosperm, full endosperm and sunken endosperm. So these two characters they are studied, therefore it is the 2-point test cross, and we have calculated the distance here. So the purple sunken was 60, purple full was 1667, white sunken was 1575 and white full 58. So when we calculate the formula to calculate the crossing over frequency is the number of recombinants divided by total number of progeny into 100. So the total number of progeny, that means to calculate the total progeny, parental as well as recombinant. So that is 3360. Okay, and out of that the recombinants are 118. So it gives the 3.5% of crossing over frequency. That means the distance between the purple and full is 3.5 Morgan unit. Morgan unit, this unit is given by the scientist T.H. Morgan, or the 3.5 centimorgan. Okay. So it is the 2-point test cross. Okay. I hope you got the point. Thank you very much.